Okay, I think it's time to get started. Good afternoon, good morning, everybody, depending on where you are. I'm Badr Benjloun, happy to meet you again in this new session of the INRA webinar series dedicated to discuss trends and innovations in agriculture. I would like to welcome all the participants from different countries for taking the time to attend this webinar. And I'm looking forward to their potential inputs and comments during the Q&A session. Before going further in this session, I would like to ask you all to please mention your full name and affiliation on the participant list. Participants who are not identified at all will be logged off in 5 minutes. Please also make sure your microphones are off through the session unless you are given the floor to speak. Thanks for your understanding. Today we are delivering the last one of the 6 webinar mini-series that started in November last year, focusing on hemp and cannabis. This series of webinars is framed in the promulgation of the law 1321 on the legal use of cannabis in Morocco. This is a new bill that marks a turning point in the use and perception of this plant and its outlets in the country. It opens several perspectives and imposes also some challenges to current and future potential actors working in the related value chains. Therefore, there is a growing need in Morocco to exchange scientific knowledge and learn from practical experiences to fully take advantage from the opportunities offered by the new law. So far, 5 speakers have taken part to this webinar series, to this mini series of webinar. They spanned a wide array of topics, their talks ranging from history and legislation, sociology, to medical and other uses of cannabis, and also agricultural aspects. Continuing along this path, I'm delighted to introduce our today's speaker, Dr. Jose CELEDON, and the topic he's going to cover. So as an introduction, Dr. Jose CELEDON has worked in plant genomics and terpene biochemistry for more than 10 years in different academic and industry labs. He currently leads the genetic team at Aurora Genomics to develop the fundamental knowledge to breed the next generation of cannabis cultivars. Dr. CELEDON will share with us his knowledge through a talk entitled Recent Advances in Cannabis Genomics and Its Implications for Breeding Programs. Before going further, I would like to mention that the main talk will take up to 40 minutes, right, Dr. CELEDON? Then we will have the Q&A session that will last around 20 minutes. Please put your questions on the chat dashboard as the presentation progresses. Participants also will be given the opportunity to ask questions or provide comments orally by raising their electronic hands during the Q&A session. Questions can be asked in English or French. Dr. CELEDON, we are looking forward to listening to you and knowing more about this exciting topic. Thanks. The floor is yours. Dr. CELEDON? Dr. Jose?
Yes, thank you very much for that kind introduction, Badr, and thanks everyone for joining this seminar today. It's really a pleasure to be speaking to you. I just want to check that the audio and the video are coming through all right, or is it a bit slow?
a bit slow, but it's okay. We can see you're moving. Yeah, it's okay.
Okay. Yes, I'm trying to minimize window that I think it's making things a little slow. If you bear with me for just a second, I'm trying
Yes.
to fix this. Okay, is it a little better now?
Yeah, I hope. Yeah, it's okay. Perfect.
All right, so thank you very much again. It's a great pleasure to be talking about cannabis. Cannabis being a new plant to me, plant that I've been working since I joined Aurora, but definitely a very interesting plant and a very intriguing plant that has puzzled people for a long time and has also served people in many different ways. And so today I'll be talking specifically about the genomics of cannabis, what we have done, a little bit about what other people have done, and the main focus is how all this information can help breeding programs. So going into the next slide, a little bit of history and about cannabis. So probably you all know that it's a dioecious plant. It's annual, which means its cycle is completed in a year, and there are female and male plants that are separate plants. What we know about cannabis is that it's been cultivated by humans since very ancient times, probably 10,000s of years. Um, Probably more for food and fiber in the beginning. And then lately, about 2,000 or 3,000 years ago, people discovered that this plant can also interact with our brains and nervous systems and has psychoactive properties. So it's truly a multi-purpose plant that has many applications and many uses, and all of them quite important. It was, however, declared illegal in many countries due to its psychoactive properties. That led to basically a complete stop or a very significant slow research in this crop. However, things changed in started changing in the early 2000s, and Canada in particular legalized the use of cannabis for medicinal purposes in 2001, and then for recreational purposes for adults in 2018. And that was a big milestone in the history of cannabis. Since then, since cannabis was legalized in Canada, it has been a very prominent actor in the economy. And between 2018, since legalization, and 2021, it contributed, it has a total amount of sales of about 11 billions, which contributed an estimated 43.5 billion to Canada's GDP, the gross domestic product, and that overall turned into 15 billions of tax revenue that the government has been able to use for different social programs, research as well, in particular in cannabis, and a number of other uses. Also, a not insignificant number, about 150,000 people, are directly employed by the cannabis industry in Canada. So this change from going from a banned illegal plant to being a legal plant that can be used for medicinal and recreational purposes has had a significant impact in the Canadian society and economy. So now more specifically in science, cannabis legalization also was a very important event. And you can see here that the research done in cannabis and cannabinoids has been going upwards very steeply since legalization in the year 2001 for medicinal use. There's a couple other very significant milestones. A little increase in the 1960s. That's approximately when the structure of THC was solved by Dr. Michelin. That triggered a number of research and a lot of research, but then slowed down when cannabis became illegal. And then you can see also more lately in the year 2018 that also there's a significant change in the slope of that number of publications. So that's been great because before we didn't know much, and you can fairly say that cannabis was a laggard, that was a plant that was ignored, that could not be researched, not ignored on purpose, but people could not do research because it was really difficult to get access to the plants, to living plants, and do experiments or studies of any kind. And that has changed dramatically since legalization. So that is great for us. It's great for many people. And I think it's great for a plant that can serve so many purposes. So that increase in research has really laid out the foundations to continue that and to catch up with the rest of the crops that we use in for food or for materials. So here you have a bit of a more detail of some of the milestones that have been achieved since research started in cannabis. So as I said, THC structure in 1964 was a very important milestone. Then in the year 95, the protein sequence of the THCA synthase was solved. Then in 2004, it took about 20 years to clone the gene. So this is before the age of high throughput sequencing. Of course, it took a long time to clone this gene, but that was also a very important event. And then after that, with the development of sequencing technologies, we have had a number of papers. The first one being in 2011 with the first draft genome and transcriptome of a drug type cannabis. And then characterization of different genes along the pathway, including genetic maps and physical maps. So these are some of the milestones. There's many other milestones, and there's many more studies that have been done in cannabis, but I wanted to highlight these because are really important for understanding the psychoactive properties of cannabis and its medicinal uses as well. And the ones that you see in this slightly different purple color or reddish color are those that we have directly, or I should say indirectly contributed through a person that was very much part of the core team at Aurora Cannabis and OCO. So Jonathan Page, probably many of you know him or have a chance to meet him. He was really a visionary in cannabis science. He saw that this plant had a lot of potential. And when it was still illegal, he managed to get access to the plant and sequence the genome in 2011. And that was really a resource and a source of information that has benefited so many academic labs and so many companies and industries as well. So John, he was the founder of Anandia Labs. Anandia Labs was acquired by Aurora, and then Aurora developed this business unit called OCO, which it's responsible for licensing the cultivars that we are producing at Aurora, which I will speak to a little bit later. So John, from the very beginning, when he founded Anandia, he had this idea of doing very basic research in cannabis, and at the same time use that basic knowledge to develop better cultivars, to do breeding in cannabis. And that's the reason why Aurora acquired Anandia and what we're still doing and the main goal of Aurora and OCO in particular. So because we had John as our chief scientific officer, and currently he's no longer our chief scientific officer, but he's still helping and in the Aurora family in a more senior advisor role. But because he was in academic units before, and he hired a team of PhDs and scientists that were very much in different academic institutions, we have established a number of collaborations with academic labs that have been really a catalyzer of the knowledge that we've been gathering at Aurora and OCO. And that knowledge is, it's been published. I'll refer to some of the highlights of that. And it's been, although very basic science, it's been tremendously helpful for breeding and developing better cultivars. So one of the first works we did was in collaboration with Lacey Samuels from the botany department at the University of British Columbia. And they had two brilliant students and postdocs that were working on cannabis trichomes. Trichomes, I guess you are mostly familiar with what they are, but they are the structures that grow in the epidermis of cannabis and also many other plants. There's different types. The ones that grow in cannabis are secretory, which means they have uh, this head on the top, which accumulates high concentrations of metabolites. In this case, cannabinoids and terpenes, and a number of other ones that are present in smaller amounts but can still play important roles. So although we knew trichomes, people knew about trichomes, they knew that THC wasn't there, a really detailed characterization of their morphology and structures was lacking. And Aurora collaborated and provided resources to fund this research, which reveals some really striking images and features of trichomes. You can see here in living trichomes that have not been fixed or stained, how the vesicles stained in that blue shifted color are traveling towards that main secretory cavity, and that's where the metabolites accumulate. And this was done by very specialized microscopy techniques that Lacey Samuel's lab has been working for a long time, and allowed us to tell the differences between the different types of trichomes in cannabis. The main type being the stalk trichomes, these ones that are sitting on a stalk of multicellular cells, are the most prominent ones in the female flower of cannabis. But there's also some other type of trichomes that are actually quite different in their absorbances and also in their structures. You can see the number of smaller vesicles and the different color speaks to the different metabolites and how they're organized inside the trichomes. You can also, one of the reasons why we studied this is because we knew that trichomes were the factories that produce the metabolites that people are considered valuable for medicinal and recreational purposes. So understanding how they're formed was very important. And up to that point, it was not known if the different types of trichomes are completely different structures or if they are related to each other. And this study showed by measuring the fluorescent properties of the trichomes and the diameters of the largest droplets in the cavity, that the sessile trichomes, the ones that are very close to the surface, or the ones that are stalked, sitting up in the multicellular stalk, are actually related to each other. So it's a developmental program that goes from a sessile trichome that then starts growing and changes the number of droplets and the absorbance of the droplets, but ultimately it ends up being a stalk trichome. And you can see that because there's a good overlap in the diameter of the droplets between the yellow dots that are the sessile trichomes and the purple dots that are the stalk trichomes in the calyces. So that overlap tells us that there's a continuous gradient, a developmental gradient that goes from sessile trichomes to stalk trichomes, and that these are actually the same trichomes that are just taking some time to develop. Another piece of information that support this developmental model is that the number of cells in the disc, the cells that are at the base of that secretory cavity, the cells that actually produce the compounds, the number of cells per trichomes are the same between the sessile trichomes and the stalk trichomes in the flowers, and those numbers of cells are different from the trichomes that are found elsewhere in the plant, including the leaves, for example, that are also having sessile trichomes, but with a different number of cells. So understanding this, it's really basic science and basic information, but from this point of view of a company that is trying to sell and produce genetics, new genetic lines that are better in quality, that have improved properties, it's very important because, for example, and some of you may know, the trichomes, the head of the trichomes can be easily detached, and trichomes can be lost from the flowers, which is a negative outcome for a consumer or for even for the grower. You don't want to be growing a plant that after you dry is actually losing the trichomes, or if the trichomes are breaking. So understanding how they're formed, the differences, it's very important to be able to select the best genotypes and use those for crosses or for further studies. So that was a really important study that Aurora supported directly with funds and with resources, and that actually turned out in a really nice collaboration with the Samuels Lab at UBC. On a different topic, we have also studied terpenes, which probably you know are small molecules from the plant produces. They are specialized metabolites that play many roles in defense mechanisms and in signaling to other plants or to insects. They sometimes serve to attract or repel insects. So they're really important, and they're widely known in many different plants. And in cannabis in particular, they accumulate also in the trichomes together with the cannabinoids and contribute to the aroma of cannabis, which is one of the features that consumers have different preferences. Some people like different types of aromas, right? So with your Bowman and Judy Booth, a PhD student in the Bowman lab, we started doing this study to really get a good understanding of how terpenes are produced and how much variation of terpenes exist in cannabis. This study revealed a number of things that were very interesting and that were not known before. For example, across different cultivars that you see in the x-axis, lemon skunk, chocolate, etc., you can see that the terpene content between leaf, a juvenile young flower, or a mature flower changes drastically, not only in amount, but also in the types of terpenes that are accumulated in the trichomes. A young flower accumulates more sesquiterpenes, which are larger terpenes with 15 carbons each. And mature flowers, by contrast, accumulate much more monoterpenes, which are smaller terpenes only with 10 carbons, more volatile, and presumed to contribute more to aroma. You can also see that there's a number of differences between cultivars, so between lines, that some of them accumulate more sesquiterpenes than others, and some of them accumulate a higher amount of total terpenes overall. So these are important features that were not well described before, and this was the big part of a study on cannabis terpenes, where we also did a transcriptome sequencing of trichome-specific RNA, and that allowed us to understand which genes of the terpene biosynthesis pathways are the ones that are contributing to these terpenes. There is hundreds of terpenes reported only in cannabis. Across plants, there's thousands of terpenes molecules that exist. And that chemical diversity in terpenes, it's really important in the quality of and the perception, not only by insects, but also by humans, of different plant attributes. So we wanted to understand the biosynthesis of these terpenes, and when we sequenced the transcriptomes, we were able to actually characterize in vitro and in planta a number of terpene syntheses in cannabis. And this shows only the monoterpene synthesis that Judy Booth was able to characterize and the products that they produce. So just to understand this plot that has a lot of information, you can see in each row there's a different terpene synthase. There's also at the bottom the different cultivars used in the study. And when you see a dot, that means that this terpene synthase, in this case, for example, TPS33, produces alpha terpenin, which is denoted in the bottom of the plot. And that would be the only terpene synthase that produces that monoterpene. And that monoterpene, it is found, although you can see a very small orange dot in all the cultivars. It's not a dominant terpene, but it's found in all of them, and it's produced only by this terpene synthase number 33. A different story, for example, would be with here with a monoterpene alcohol that we could not identify with standards, but it is found only in 2 cultivars. And in this study, we did not find a terpene synthase that produced that monoterpene alcohol. So it tells you, this plot tells you that there's a number of terpene syntheses in cannabis that contribute to terpenes. There's many more that are still to be characterized. That's why you see some areas of this plot that have no enzyme that produces that monoterpene. However, it is found in all the plants that we studied. So this was the beginning and laying the foundations for understanding terpenes with a particular focus and application of improving aromas, which is one of the goals that we are working towards at Aurora OCO. So please feel free to interrupt if there's any questions. I cannot see the hands up, so please feel free to just open the mic and ask any questions if there's something you would like to ask. So now in a more current...
It's okay. Yeah.
Okay, excellent. Thanks.
Okay.
Thanks so much.
Thanks.
Okay. So those 2 studies that we did with different labs at UBC were studies that are already completed, and we were extremely happy. So we decided to go and explore a new area. And this was, those 2 studies were done in the very, very early days of cannabis legalization. So things were a little bit more difficult to share materials, to have people come and go. Everything had to be with alarms, and access to the plants was very restricted. It could be done, but it was not necessarily easy. And we were lucky that actually one of our facilities, it is on UBC campus. So we have labs and ability to grow plants and do experiments on UBC. And that's why UBC labs can come to our labs and actually have access to materials. So more recently, almost 2 years ago, we also approached the RISPER lab at UBC with Lauren Risberg and Marco Tedesco to study quite a growing problem in cannabis. And that has to do with the diseases that affect the crop. So before cannabis was legal, people used to grow cannabis in small scale operations, sometimes in the basement or sometimes in different places. And it's probably they controlled, if there was any disease outbreak, they probably use fungicides, and that basically solved the problem for them. And there was very little regulatory control of any unauthorized products in cannabis. Everything was in the black market. Now, however, after legalization, there was a number of strict regulations that were put in place. There's a number of tests that you have to do before you can release any product into the market. And also the growing conditions were different. Instead of being small, we are growing now in very large scale facilities where you have lots of plants. It's more like a monoculture, and any disease that appears can spread very quickly. There is and there's no products that can be used to control diseases. So fungal diseases in particular are a big problem. So we decided to develop a pan-genome and a genome-wide association population that we could use to develop genetic markers for breeding, in particular for diseases, for finding resistant to diseases. So the RISPER lab and the Tedesco lab together with Aurora, we put together a cannabis diversity or CAND population that was going to be used for phenotyping disease resistance, and also sequence, so that we could know the genetic makeup of cannabis in a very diverse population. And then at the same time, we decided to sequence the genome, the full genome of 12 genotypes that represented that diversity, and that could be used for developing markers. So just to give you an idea of what that CAND population looks like, our diversity panel is extremely diverse. This is just a subset of the plants that we they vary widely in their color, their architecture, their flower structures, their cannabinoids, of course, terpenes, and their ability to become infected by different diseases. So it's a population of more than 500 different drug-type cannabis lines that we keep alive. So it represents a germplasm resource for us for breeding, for accessing rare alleles or plants that have traits that are beneficial. We did whole genome resequencing at a 30x coverage for calling SNPs and identifying genetic variants that could be associated with traits of interest. And these, so far, we have obtained a SNP dataset of more than 7 million high-quality SNPs. So when it started screening for disease resistance, in particular for powdery mildew resistance, which was the goal of this project, to find a marker for powdery mildew resistance, we have found so far a very promising marker that we are working to introduce into our elite cultivars so that we can develop not only a plant that has high yield and appropriate profiles of cannabinoids and terpenes, but also that can grow disease-free. And we think that this is going to be a very important and valuable trait for growers, that is going to reduce the cost, the labor that is used in order to control powdery mildew in cannabis. It's pretty significant, and we're looking forward to being able to release new cultivars with these properties. So I've mentioned already a couple of times that at OCO and Aurora, we have a strong focus on breeding cannabis, better cannabis cultivars. So I'd like to speak now to that part of the research that we do. And this is something we do not through academic collaborations. This is something we do in-house at our facilities. And I'll just like to explain why it is important. So one thing that we should, that you probably know, but it's good to remind everyone, is that cannabis is a clonally propagated crop. And a clonally propagated crop has a very high cost for production. In order to fill a room, or imagine even if it is an outdoor growth, you will need to prepare the clones manually, root the clones, transplant the clones, and then bring them to the field. A much cheaper way to do this would be using a seed propagated crop. And ultimately, that's the goal we have in our breeding program. So why new genetics? Well, there's a number of reasons. One is what I've mentioned a couple of times, is that there are production challenges, including disease, the cost of labor, producing the plants and putting plants in the greenhouse is very expensive. There's also some market trends that are changing. They are dynamic. People, it's like to try different things. And there's also, since cannabis is legal since very recently, there may be new sectors of the population that perhaps have different preferences to it, that will join or become consumers or users in some time. And then, as a company in particular, everybody would like to be differentiated, to have a product that is unique, that people can relate to, that you can have as a good cultivar. Sometimes also what happens is that you can lose a cultivar due to disease or other reasons, and in that case you need to replace it. So for all these reasons, we think that having a breeding program is a key component of a successful cannabis business. So for a long time, the way people have been providing new genetics for growers is through a process called phenohunting, which basically is you make a cross, you germinate the seeds from that cross, and you have to grow the plants to maturity and identify those that are valuable and that have the attributes you're looking for. As you can imagine, this is a slow, it's also very, very expensive to do. And and then not very efficient. You have to grow lots of plants to find perhaps 2 or 3 that have the appropriate qualities for becoming a successful cultivar. So, and this is because the chances are very small. When you have the cannabis genome has about 30,000 genes, and having the right combinations of all those 30,000 genes, it's a really low probability game. And we think we are working towards applying, and we are applying molecular markers for speeding up and making this process more more efficient, and less random. So for that, we have established a number of research facilities. Our core research labs are in Vancouver and Comox in Vancouver Island, where we have our breeding and genetics in Comox, and the genomics and metabolomics in Vancouver. Whistler and Toronto are also involved in our research activities in different capacities. And together we have developed a pipeline that is quite powerful in producing high quality cultivars. So this is the facility in Comox where we do the selections and the crosses. It's really a beautiful place. It was designed from the ground up for breeding cannabis. It's got all the features that you would dream of in terms of breeding, and it was built purposely for breeding cannabis. So here's where we maintain our germplasm. Here's where we have developed where we keep our cultivars, where we make our crosses, and we can do really extensive and detailed phenotyping of plants to look for new traits, for existing traits that have room for improvement. And it's a place that if you ever come to Canada and to Vancouver, I do invite you to take the trip to Vancouver Island to visit this place. It's a wonderful facility, and you will see some amazing diversity of cannabis. Of course, we have a number of labs that are where we perform the analysis and of our plants and our process. So it's a very beautiful place. We have a nice variety of teams that are dedicated to different parts of the breeding pipeline. We are able to do, in combination with the Vancouver labs that I lead, to still perform basic and foundational research that allows us to develop better cultivars. We have our own testing labs for disease and pathogens. We have our own analytical labs to study the chemical profiles of these plants. And we also have capacity to do sensory science. So this together, I think, covers the most important angles that are required for having a successful breeding program. So, and the main purpose is to apply molecular markers so that we can really germinate thousands of seeds, and at the seedling stage select those winners, those plants that are going to have the right morphology, the right flower quality, the right terpene profiles, the right cannabinoid profiles. So with all that information and with all this support from our teams and people, we've been developing cultivars for a long time, and we have come up with a pipeline that allows us to do this at a large scale. So in phase 1, when we start our breeding cycle, we have a large number of different genotypes that are selected based on plant shape and potency. The ones that are selected and passed through the cycle to the phase 2 or cycle 2 are then further selected for potency, but then in addition to potency and terpenes, we include yield and so forth. You see that the number of genotypes is being reduced, so only the winners pass to the next phase. And as we move down in that pyramid, you see that the number of clones or the replicates that we use, it's much higher. So that gives us an increased confidence in the data we're collecting, and that the phenotypes we've been selecting are repeatable. So that's what we are doing for clonally propagated cannabis. At the same time, we're also working in a true hybrid breeding scheme where we think the future of cannabis is not going to be a clonally propagated crop, but it's going to be a seed propagated crop. And for that, we're performing direct process and inbreeding so that we can, and then testing those hybrids to pick out the best performing ones. So I should say that in cannabis, as opposed to other crops, you do need to have a testing and analytics lab that provides you accurate information on each of those selections and lines that you'd be moving forward in your breeding program, either for inbreeding or hybrid seed testing or for clonally propagated lines. And this is really a centerpiece of our research. Then as cultivars make it towards the end of the pipeline, we also evaluate their sensory attributes, because even though a plant can have very high yield and be highly resistant to diseases, if it doesn't have the sensory attributes that people are looking for, it will not be selling very well. So we have developed a number of qualitative and semi-quantitative sensory analysis that we're using to compare the different genotypes that we have in our pipeline. So some of the lines that we've been producing have had a very, very good cannabinoid profiles and terpene profiles. Also, you can see that the flower quality, it's really great. And this is just a sample of the novel genotypes that make it to the final line in our breeding program. Different colors, different THC concentrations, and different aromas. So OCO and Aurora together have a really strong track record. So far, we have screened more than 10,000 unique lines, which turn into more than 13 commercialized cultivars. And this is internally for Aurora and also for other cannabis companies that also license our cultivars. Of those 13, we have 5 so far that have top reviews that have been highly highlighted in different social medias and competitions. So overall, we have achieved an increase in THC, terpenes, and yield, which are very important attributes for growers and consumers alike. And the next milestone, the next goal we are working towards is to achieve the same quality, but with powdery mildew resistance. So, and although this is for clonally propagated plants, in the future, we do see the cannabis industry being very different. Probably we would like to see cannabis being grown outdoors. We're working towards that. And that will be reduce the cost of production tremendously. Currently, cannabis has a very high carbon footprint because greenhouses have to be heated. They need artificial light, and we do have free sunlight if we were to grow cannabis outside. So that would be a tremendous advantage. It would also be good for the environment. So that's how we see the future. And I think for moving outdoors and for moving to a large scale normal agriculture crop, seed propagation, it's going to be probably very important. So with that, I'd like to conclude and see if the audience has any questions or comments or that I'll be happy to take.
Thanks, Jose, for this very nice and rich talk. Give us a great ideas about your experience mainly at Aurora by taking advantage of what has been done from fundamental science to apply this in breeding of this plant. So thanks for this rich talk. So I will take questions from the chat. Okay. We have one question from Dr. Abdelfatah Barrada. He's asking, do you work on marijuana cultivars only, or do you also breed for CBD and other cannabinoids?
We also breed for other cannabinoids. Yes, that's correct. Medical users have yeah, an interest in CBD cultivars as well. So these are still high producing CBD cultivars, but it's not only THC. Yeah.
Okay. Waiting from others to put their questions. I have a question. So, as I understood, you are using marker assisted selection to select the
Yes.
to make selection. So I guess this is linked to some traits that are controlled by some major genes. Not I'm talking about the traits that are controlled by many genes with small effects and so on. Yeah, the difference. Are you planning to use genomic selection, not only marker selected selection, marker assisted selection? Sorry.
Yeah, so genomic selection, it's a very interesting approach to do breeding, and I think it's a bit early to say if it's going to work or not, but we're definitely keeping a very good eye and using some of our data sets and populations to test this idea. It would be great if it can work. It can definitely help you with these multigenic traits, which cannabis, there's a few that are very important. So we are exploring that possibility, but I cannot say much more than that. It is definitely something that has worked well in some plants. It's definitely been used for animal breeding. But in plants, I know also of a few examples where the results have not been as good as expected. So the effort that you have to do to try to really implement this, it's not insignificant. And if you don't see the outcomes that you were hoping for, then basically you have used a lot of time and money to do something that didn't work. So we are a bit cautious, but we are definitely exploring and keeping an eye on the technology and what other people have been able to do with other plants as well.
Mm-hmm. Okay. We'll take the next question from Zakaria Kamal Dori. What's the best ways to test the THC or the CBD efficacy, effectiveness upon a laboratory?
The
He's asking
efficacy.
about the effectiveness of THC or CBD.
So that I guess so I'll take the question as meaning perhaps in the how it is the effect on people, right? And that is really an unexplored area. So that if you want to test THC or CBD in the laboratory is very straightforward. There's a number of techniques you can use. So I don't think the question refers to that. We use HPLC, but there's a number of other methods that can be used, right? So LC-MS or even near infrared spectrometry can also be used for testing and for measuring these compounds. But the efficacy or the effects on people are much more difficult, and that is because it's extremely subjective, and it's also you need basically some kind of, in Canada at least, that will be like a clinical trial where you have to get ethics approval, et cetera, if you want to measure the effects of cannabis in people. So that is, it's actually nothing very trivial to do, and it's so subjective that you will probably need to do a large trial in order to get an idea. Let's say that this cultivar has an energizing effect, or this other cultivar has THC and CBD, but also a relaxing effect. Then if you want to test that, you will need probably large numbers and surveys to ask how people felt the effects. So if that was the intent of the question, I think it's something that I don't have the answer. I don't know what's the best way to do it. People do report different effects, right? So it is true. The challenge is that it's a very difficult variable to measure.
Okay. Next question from Abdel Fattah Barrada. How is the cannabis market doing in Canada? In Colorado, there has been a lot of challenges to the marijuana market.
Yeah, so I think it's not my area of expertise, but I'm definitely familiar with the news and what you can read in the newspaper and some of the internal conversations. It's the market. It's been, I guess, unexpected. We thought people in Canada thought that the illegal market will disappear very quickly, and that consumers would buy legal cannabis in a short time. And that has not been the case. So many of the predictions of volume, how much cannabis we will need to produce and how much cannabis we will need to sell, have not been according to the predictions, which has meant that many cannabis companies, including ourselves, have had to close some production facilities. And now I would say that we're finally reaching a balance between supply and demand. The illegal market, I believe, is now a bit less than 50% of the total cannabis consumption, but close to 50% is still a lot if you consider that recreational use was legalized 4 years ago, more than 4 years ago. So it's taking more time than anticipated, and there is a lot that I think politicians and different government agencies have learned about this big experiment about legalizing cannabis in Canada. But it's unfortunately it's not. That's all I can say about the market.
There is a comment from Zakaria Haman who asked the question about the efficacy. Yes, he said, yes, that's exactly my question. I think we will need many legal documentation in order to start studying the clinical effects of the plant, because it might be clinical to the patient if not used properly. Okay.
Yeah, there's some interesting approaches to this. There's a company called Leafly that basically collects user data that people so they have all the cultivars that you can find in the market, and people can show their preferences, and their preferences are somewhat related to the effects they experience, right? So it's a way of collecting some information about the effect. But if you want to do a controlled clinical study where you have placebos and double-blinded, et cetera, so that you can really account for the effects of the plant and the cannabinoids, then that becomes a lot more challenging to do. And currently, I know there has been changes in the regulatory space in Canada, and some things have become a bit more flexible, but some other aspects of research have become actually a bit more limiting. So we are learning on how to do research on this plant, and it's something that, for example, I don't think there was any such studies, for example, for the wine industry, right, or for the beer industry. I think people just have focus groups, perhaps, where you can invite people to participate in a focus group where they tell you in a small number, they tell you what they think about a product. So that is something that can be done. But a different story is a double-blinded or controlled study where you actually measure the effects by asking questions and a survey, right?
Okay. Next question from Brahim A. Talib. He has a question about feminized cannabis plants. When he practices the feminization on deciduous cannabis plant, will he have descendants on plant after? He's talking about seeds. If yes, will the plant become monoecious at 100%? If not, what will be the ratio between male and female plants?
Yeah, if you have a female plant and you induce the production of male flowers, that's well described in the literature, then you can make a cross between you can make a cross between two female plants, and that's a very unique feature of cannabis, that you can induce the production of pollen or male flowers in a female plant, and that allows you to produce what people call feminized seed, which should be in theory female plants only. Now, cannabis is a plant that can be stressed easily, and sometimes stress or different environmental conditions can induce the formation of male flowers in female plants. So that's something that people describe as hermaphroditism. So a female plant can also become hermaphrodite or having both types of flowers. And that's something that cannot be excluded, right? Even though if you have a feminized population or seed lot, you can still see some male flowers being produced.
Okay. Abdulfatah Barada is asking about your contact information. Can we share your email address or something like this in the chat? It's okay?
You know, I guess it would be fine. So you can contact me on LinkedIn. If you find me on LinkedIn, definitely maybe that would be better, because my inbox is already full every day, so I'm a bit hesitant with my email address. But LinkedIn works. I'll keep an eye, and then I can share my email address with people through after a LinkedIn contact. So definitely just make a reference to this seminar, and I'll respond to you, and then we can exchange emails and start a conversation via email. That would be fine with me.
Okay. Abdulfatah Barada is a scientist at Colorado University. He contributed to this mini series by a talk. He was the first talk. Okay.
Oh, okay.
The question from Anas Khaldoun. My question is about the molecular markers of THC synthase and CBD synthase. There is anyone for these enzymes, and are these enzymes similar in all cultivars, and how we can know which one is dominant?
Yeah, so that's well studied. I don't remember the year of the paper, but the synthesis, the cannabinoid syntheses are well described. And when you have a non-functional THC synthase, then you have a plant that produces CBD. It's a CBD dominant plant. When you have both syntheses being functional, you have what people call a balanced chemotype. So different ratios, but approximately 1-to-1, or it has this a variation too. It's not 1-to-1 exactly, but approximately close to each other, CBD to THC. And then when you have only a functional THC synthase, you have a THC dominant plant. So these are genes that exist in several copies in the genome, and some of them are non-functional at all. They just exist in the genome. They're still not fully understood whether they just lost the function, they never what they do when. But the syntheses, in particular, the THC synthase and the CBD synthase, there's markers for that, and they are disclosed in publications. You can definitely screen for that with marker-assisted selection. Mm-hmm.
Okay. I think there's no further questions. Okay. I think we are getting now to complete. Okay. So we are moving towards the end of this webinar. I would like to mention that there is survey links on the chat dashboard made by my colleagues. Please the audience to use it to provide us with your assessment of this mini series. and also the INRA webinar series. It will be helpful for future improvements. You can also request your certificate of attendance through this link. Finally, on behalf of the organizers, I would like to thank very much our guest speaker, the last speaker of this mini-series of webinar, Dr. Jose CELEDON, for his time and availability in the preparation of today's webinar, and for sharing with us his deep knowledge. My thanks go also to all the participants and the organizing committee, especially to my colleagues, Bouteyna Abidou, Atman Sabatar, Dati Razin, Uddin Bahri, Abdelaziz Yasri, and all the scientific committee of the INRA webinar series for their crucial contribution in the organization of this series. At the end of this series, I would like to thank warmly all of the speakers who contributed to it, namely Dr. Abdel Fattah Barrada from the University of Colorado, the US, Dr. Abdelaziz Yasri from INRA, Morocco, Dr. Abdel Fattah Marzouki from the University Abdel Malik Saadi in Morocco, Dr. Khalid Mouna from the University Moulay Ismail in Morocco, Carla Gage from the Southern Illinois University, and also the last one, Dr. Jose CELEDON. I would like also to thank all the moderators of these 6 sessions, mainly Dr. Iman Tamil Alami, Dr. Mohamed Boughlala, and Dr. Adil Sariwi. And finally, I would like to thank Dr. Fawzi Bkawi, Director of INRA, for all his input into this series, and especially into this mini-series on hemp cannabis. Dr. CELEDON, would you like to say something, the last word?
Well, thank you very much for the invitation. It's been a pleasure to contribute to this mini series of seminars. And as you start your path in the cannabis industry, please reach out if you have any questions, or if you are looking into doing any kind of research. As a company, we are actively doing a number of projects, and I'm sure there will be some overlaps and some common interest. So yeah, just to extend an invitation to reach out and to keep in touch between INRA and Aurora Oko. We love to do research, and we think cannabis is a extremely interesting plant. So it would be great to find an equally passionate team of people that are also doing research in cannabis. So please let us know what you find out, and we'll be in touch as well.
Yeah, perfect. Thank you very much. So looking forward to meet you in another webinar. Okay. Bye-bye.
Absolutely. Have a
Bye,
good day.
everyone.
Have a good evening. Bye.
Have a good day. Good evening. Bye.